So here's three examples of nutritional labels, and we're going to focus in on the fat content on each of these labels, just as some examples. And we can see here that on the left we have an egg, in the middle that's ground beef, and on the right we have from a soda pop. And you can see here that, as we talked about before, the total fat in soda around in there, you can see there's no fat in soda. But if you look here in ground beef, there definitely is some fat. And although at this point in this course we've only talked about fatty acids, there are other molecules that would also be included in total fat. And we'll talk about some of that. But a lot of the fat that we get is in the form of fatty acid, but not necessarily all of it. And you can see here for the ground beef that the total fat is 9 grams per serving, and the amount of saturated fat is 2.5 grams, which would kind of give you a sense that the unsaturated fats would be the remainder of that, up to 9 grams, just as a ballpark figure. And then over here with egg, they also show, and this is not always shown, but you can see here, there's their fat. They have total fat at the top, they have saturated fat, and they also show the amount of trans fats. And you can see that eggs don't have any trans fats, but they would have cis fats, but they don't explicitly show that. So nutritional labels don't show every type of fat that's in the product, but it does give a certain rundown of that. We also talked about omega-3 fatty acids in class, and here are 3 fatty acids. Now, fatty acids actually have a common name. We don't need to memorize these names. We learned a different way of naming them. But you can see here that there's linoleic acids, very common natural fatty acid, and a couple other different fatty acids. And notice that some of them, a lot of them in their names will have oic acid because it is a carboxylic acid. And so some of them will have that oic acid as part of their name, but that's just the organic nomenclature that we learned already. Now notice all 3 of these are omega-3 fatty acids. If you look at the numbering that they did, in blue is the counting from the delta end, and remember the delta end is the end where the carboxylic acid group sits, and the omega end is the opposite end of the molecule. And they did numbering for both the delta and the omega. The delta end numbering is in blue, and the omega numbering is in red. And you can see here that if you look at the third carbon from the omega end, so highlighted with the red 3, you can see that there is a double bond between the third and the 4th carbon. And you can see that in all 3 of these structures. So all 3 of them are omega-3 fatty acids. The other thing to know about omega-3 fatty acids, when we talk about them for nutritional purposes, omega-3 fatty acids are always in cis conformation because that's the natural version of them. Trans aren't seen too often naturally, but cis are seen all the time. So the healthy omega-3 fatty acids will always pretty much be in cis. Trans fatty acids, even if they're omega-3 trans fatty acids, are not really a healthy molecule. It's these cis ones. But when you talk about fish oils, things like that, those are all going to be, even though only is typically referred to as omega-3 fatty acids, they're really cis omega-3 fatty acids. And you can see that there in their structures that all of them are cis. Not only are they omega-3 fatty acids, but some of them are also omega-6. As you can see, the red 6 on these structures also shows that they have a double bond between the 6th and 7th carbons, counting from the omega end, and there's even some omega-9 fatty acids. And each of them have a different level of nutrition for us and healthy nutrition for us. So now we want to go ahead and now use our understanding of fatty acids and kind of look at now families of lipids. And so we're going to look at 6 different families of lipids. And a couple things that you want to be able to, or a couple things you should focus on in the next few slides are, are, first of all, knowing that there are 6 families and what their names are, as shown here on this slide. You also want to be able to identify structures of these. So if I showed you a structure of a lipid, you could classify it as one of the 6. You don't have to be able to draw them, you just have to be able to visualize them and look at them and be able to recognize which of these 6 it is. And we'll talk about features of each of these that will help you with that. Then the last thing you want to know is what are the biological roles of each of these. So be able to identify them from the structure, and then also be able to identify what is a functional role or a biological role of each of these. So let's take a look at the first family. In the first family, we'll talk about our waxes. So waxes are coated on plant stems, on fruit. You all know that if you pick up an apple, it kind of has that waxy coat to it. Skin and fur also typically has lots of waxes associated with them. And what waxes are comprised of, or how they're actually made, are a reaction, as we can see down here at the bottom, where if you take a carboxylic acid, and this is a reaction we did not learn in the organic section of the course, but now we can add it in. But if you take any carboxylic acid and take an alcohol, they can be combined together to form an ester. So just a couple of reminders of what these molecules look like. We remember carboxylic acids have that C double bond O with the OH on the end, and then an R group, meaning a long carbon chain. And these are typically not short chain carboxylic acids. Waxes are formed from long chains, so the carbon chains are very long. And then alcohols are this structure in general, where you have some carbon chain and it has an OH group sitting on it. And then just a reminder, an ester has that carbonyl carbon, and then on one side there's an oxygen with an R group, and on the other side there is another R group, and so we form an ester. And identifying esters is going to be very important in lipids because a lot of lipids have ester bonds in them, and so I'll be pointing those out quite a bit. And of course the other product of this reaction is a molecule of water. And so we can see up here, as an example of a wax, notice a couple of things. So on this molecule on the left, notice that it has 15 carbons hanging off of that carbonyl carbon there. So that's a long carbon chain, right? And you can see down here that these are formed, down here is where I'm looking at the bottom here, these are formed using long chain fatty acids. And so the fatty acid here would have been 16 carbons long. The alcohol, notice it's also formed from long chain alcohols. The alcohol's on the other side, and notice there's 30 carbons there. So you have these super long carbon chains with hydrogens all over them. Those are very nonpolar groups. So this 30 carbons with 61 hydrogens, that's very nonpolar. And that group out there is very nonpolar. In the middle between there, it's very polar. So you have this little bit of polar character and these two huge nonpolar groups on each side of that polar area. We can see that again in the structure on the right here. Again, you can see here the alcohol out here was formed by this carbon right here. So that's one carbon. There's 28 CH2s then. And then there's one more carbon right there. So that's a total of 30 carbons right there. And then on this side, there are 15 carbons on that side. These two structures tend to be very similar, but once again, what's highlighted here highlighted in yellow are the nonpolar areas, and then you have this little polar group in the middle. Understand as well, if you were to draw this out, it makes it look like the way they have it in such a condensed drawing, it makes it look like there is a pretty big polar area. But it really is not, because if I draw this structure, and now I'm going to go ahead and start adding carbons on, you can see I could start adding these carbons on, and all of a sudden this molecule gets very big. And what I drew on the right-hand side is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. This is only 12 carbons long on this side, and on the other side we got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 14 on this side. Notice in these structures down here that were given, there is a total of 15 on one side and 30 on the other side. So even this one would be a very small chain or very small wax because of that doesn't have a ton of carbons in it. But notice again here you have this big group that I'm squaring in as nonpolar, and this big group is nonpolar. Then you have this little polar area right in the middle. Now this slide just highlights the product of the esterification reaction. The esterification reaction is from the long-chain carboxylic acid and the long-chain alcohol. So we call that an esterification reaction because we're making an ester in the process. And you can see down here in the structure highlighted in blue is the part that the alcohol came from. And so you can see this oxygen kind of breaking up the two carbon chains. On one side of the oxygen is the alcohol group, and on the other side in red is what came from the carboxylic acid group, and we can really tell that came from the carboxylic acid because alcohols don't have a C double bond O in them, but carboxylic acids do. And this is the structure we find that comes from beeswax, is what we've been looking at up to this point. Now the next structure here that we're going to talk about is the triacylglycerols. Triacylglycerols are important this is the main way in which we store energy in our body. So this is, when we talk about storing up fat in our body, this is the form that it takes on. So because it is stored up in fat in our body, we also recognize that the fat in our body also serves as padding and insulation for us, so it's very good for those purposes. And these are also formed by esterification reactions. So the same reaction we just learned for waxes is actually the exact same reaction we learned for triacylglycerols as well. But in this case, the alcohol that's involved is not a long chain alcohol. It's actually just an alcohol with 3 carbons on it. And so if I were to draw that structure, let's see if I have enough space to, so it might get a little messy in here. But so a glycerol molecule, which is is where the alcohol group is going to come from, is a 3-carbon chain with hydroxyl groups attached to it, 3 of them in fact. And then the rest of the bonds to carbon that are not shown would all be hydrogens. I don't have enough space to put all those H's on there. But what a glycerol molecule is 3 carbons with 3 hydroxyl groups, 1 on each of the carbons as shown here. And so now you have the alcohol group. In fact, you have 3 alcohol groups on this 1 glycerol molecule. And what happens is an esterification reactions happens where a carboxylic acid, and it could be any fatty acid, can form an ester with any of these 3 hydroxyl groups. And what happens is in fact, in this reaction, or to form a triacylglycerol, you get 3 fatty acids that actually react with these 3 hydroxyl groups to form 3 ester bonds. And so in the structure over here, I'm going to go ahead and get rid of this glycerol molecules. And what happens here, what you can see here is that I can see an ester right there, I can see an ester right there, and I can see an ester, the central portion of the ester right there. And notice on one side of these 3 esters that are all connected, there's my original 3-carbon glycerol backbone. So that side, not highlighted in yellow, is all of the glycerol backbone. And then the other side, or at least that's where the glycerol backbone is, and on the other side is where the carboxylic acids, or in our case the fatty acids, came from. And you can see here that we have 3 different fatty acids that attached on or formed these ester bonds with these 3 alcohol groups. And so notice one thing about triacylglycerols you should recognize is that it doesn't have to be the same fatty acid on all three of them. It could be, but it doesn't have to be. And so, but nonetheless, you notice these are also long chain fatty acids. We can see highlighted here in yellow, again, very nonpolar. And so you have this large nonpolar group. Notice the first fatty acid here on top. That one has highlighted in yellow. That's 15 carbons. In the second one down, I see 8, 9, 16, 17 carbons it looks like. And on the last one down, it looks like it's 17 more carbons. The one here in the middle is an unsaturated fatty acid because I can see the carbon-carbon double bond right in the middle of it. The other two are saturated fatty acids. In this molecule that's not highlighted in yellow, those are the that's sort of the polar end of the molecule. So once again, here's another lipid with this huge nonpolar region and this little polar region. We can look down below and you can kind of see the relative sizes of sort of a triacylglycerol, polar and nonpolar. Once again, I see my 3-carbon glycerol backbone. I see my 3 ester bonds right down the middle. And then way over here, all this stuff, there are my 3 nonpolar fatty acids that were attached on through esterification to this glycerol backbone. And so notice one thing to help you out when you're looking at these different molecules is if you see, notice the name of this class of molecules is, or family is the tri, meaning 3, acylglycerols. So it tells you tri, meaning 3, and glycerol kind of tells you the backbone. And because there's 3 of them, you'll find 3 ester bonds. Notice in waxes there was only 1 ester bond in the molecule. In triacylglycerides there are 3 ester bonds. And that piece of information will separate these 2 classes or families from all the other ones. If you see 3 ester bonds, that is a triacylglyceride. If you see 1 ester bond, that's probably going to 1 ester bond, no other types, that's going to be a wax. This slide right here just highlights how a triacylglycerol is made. Basically, I talked about it on the previous slide, but just to highlight it in a single slide, it's shown here. Notice way on the left here we have our glycerol molecule. We have in the second sort of row there where it looks lined up, we have 3 fatty acids, and those fatty acids don't need to be the same. So notice to make a triacylglycerol, we basically need 4 molecules. We need the glycerol molecule and fatty acid number 1, fatty acid number 2, and fatty acid number 3. And when they come together, notice they make 3 ester bonds. And if we looked at the general structure of this, this is what it looks like. It looks like it's a glycerol molecule attached to 3 fatty acids. Kind of gives the generic or the summarized version of this molecule, where a wax is going to be a fatty acid, which I'll just highlight as Fa, connected to an alcohol. So that's sort of the difference between those 2 classes or families of molecules. The third family of lipids are the glycerophospholipids. This class, or this family of molecules or lipids, also has a glycerol backbone. And we can see that right here. There's sort of our glycerol backbone. There's our 3-carbon backbone, and which were originally alcohol groups that reacted. 2 of the alcohol groups attached onto fatty acids to form this ester bond in there. I'm going to circle these ester bonds, and sorry it's getting a little messy in there. But notice the third one didn't make an ester bond. I don't see the C double bond O in here. In fact, what I see is a phosphate in there. And so glycerophospholipids have, as one of their features, a phosphate in them. Notice in the name, glycerol means it has a glycerol backbone. Phospho means it has a phosphate in it. So if it has a glycerol backbone and a phosphate, that's a good indication that it's a glycerophospholipid. Notice what's here as well. In this, we can look at another structure here on the right-hand side. Notice, yes, I see the glycerol backbone, but I only see 2 ester bonds. not 3. I don't see the third one. Remember, an ester bond, you're looking for that sort of structure where you have a couple of R groups on each flanking this C double bond O, C single bonded to O structure. And I can see the C double bond O here again and here, but I don't see a third one. Remember, if I see a third one with that glycerol backbone, that's a triacylglyceride. If I only see 2 with a glycerol backbone and there's a phosphate in there, now we have a glycerophospholipid. So these are the things I want you to be able to do when you evaluate structures, is to find these little components and then kind of recognize which one we're staring at. These glycerophospholipids are important in membranes. So these are membrane lipids. I mentioned a couple of the roles of lipids in our bodies, in humans, actually in all mammals and plants, is that one of their roles of lipids is membranes. So waxes aren't comprised of aren't part of membranes. Triacylglycerides aren't part of membranes, but glycerophospholipids are part of membranes. Now, they're good in membranes because of the structure that they make. And what we see there is, because it only attached on 2 fatty acids, we have 2 we have 2 long carbon-hydrogen tails on a glycerophospholipid, because we only had 2 fatty acids attached on. The rest of the molecule, that glycerol backbone, that phosphate group, and a polar group, comprise the polar region. So it has this big old polar region, or a bigger polar region. And then they have these 2 long fatty acid tails. And so this sort of structure, where it has a polar region and 2 long fatty acid, or 2 long carbon-hydrogen tails, these non-polar hydrophobic tails, this really is the structure that we see in membranes all the time. So any lipid that makes that kind of structure, we find in membranes. Remember, a triacylglyceride is sort of a polar head, that's the glycerol group, and then it had not 2, and this is my quick drawing of a fatty acid tail, it had 3 of these long hydrophobic non-polar tails, not 2. And so that structure is not used in membranes. It doesn't fit well, it doesn't work well in a membrane, and so it's not. That's used to store energy, that structure. But these, where they have 1 polar region and these 2 long hydrophobic tails, great for membranes. Now, the 4th family is Sphingomyelin. And for this one, we have to remember what an amide is. And just to remind you, an amide has the C double bond O, but different than an ester, that carbon's not attached to an oxygen, but rather a nitrogen. And so in this structure, that's an amide structure. And finding that amide structure is very important. So at this point, notice we've had 2 organic families that have been kind of prevalent here in terms of identifying lipid families, and that is the ester. So finding esters is very important, and finding amides is very important. If you see an amide linkage, then you're looking at something that's not it's not one of the first 3 classes that we have already looked at. Notice the first 3 classes had esters but didn't have any amides. I never mentioned amides because there isn't any. Now we see our first amide. A couple things. This is one of the harder ones to identify because, yes, it has a phosphate group, and when you see that phosphate group, you automatically start thinking glycerol phosphate. But remember, to be glycerol phosphate, it has to have the phosphate group and 2 esters. And in this structure, you actually don't see any esters. There's none here. And so therefore, it's not a glycerol backbone. It doesn't have the glycerol structure in it. And so what this one has is not a glycerol backbone, but it has what's called a sphingosine backbone. In a sphingosine backbone, it has kind of what looks like those 3 carbons. We can see them numbered right down here, 1, 2, and 3. But notice the second carbon is not attached to an oxygen, it's attached to a nitrogen. And so that's kind of the giveaway. And that nitrogen eventually forms when a fatty acid attaches on it, forms this, what I'm squaring now, forms that amide linkage. And now we have the fatty acid tail hanging off in red there to the left of what I just squared up. The sphingosine backbone is actually a long carbon chain. It's only numbered to 4 here, but notice the carbon chain continues, as my arrow shows, in that direction. And so what you get from sphingomyelin is you do get the phosphate group, and you do get a fatty acid attached onto it, shown in red. But you also get the sphingosine backbone is not 3 carbons long, but it is about, I think it's, 4, 5, 6, 18 carbons long. 4 of them are numbered, and the rest of them are down here. And so what you end up getting from a sphingosine backbone is one long sort of fatty acid looking tail. It's not a fatty acid, but it is a long carbon hydrogen chain, just like a fatty acid is. So you get that chain, and you also get the fatty acid chain shown in red that was attached on through amide formation as opposed to esterification. So it was put on just slightly differently, where a carboxylic acid attaches to an amine to form an amide. So to form that amide, if I just round it out, it's a carboxylic acid, which is the fatty acid, plus the amine, which comes from the sphingosine backbone to form an amide. And so that kind of gives you the general where that all is coming from. Now, just as we saw before, you still get the polar head structure shown right here, and you get the 2 long chains of carbon and hydrogens down here. So again, sphingomyelin is really important in membrane biology because it has that same structure, a polar region and these 2 long chains. Now, one of those chains is from the fatty acid that attached on to form the amide, and the other one is from the sphingosine backbone. So that's where they're coming from, but in the end, it's still these long hydrophobic carbon-hydrogen tails. These sphingomyelins really almost specifically used for or in the membranes of neurons. So when people start discussing neurons and membranes in neurons, they're typically talking about sphingomyelin. So these are kind of a specific lipid that exists or used in membrane structure for neurons or nerve tissue. And we can see here, on the left-hand side of this schematic, you can see the structural details of this polar region and nonpolar region. And I'll just highlight a couple things to help you identify this as being sphingomyelin. One is, I see the polar group, or the phosphate group right there. So that helps me identify it as it can't be a triacylglyceride, glycerol, it can't be a wax. Those guys didn't have phosphates in them. And notice I see right here, kind of hiding, but there's the amide group. Notice there's the nitrogen connected to the carbon, which is double bonded to oxygen. And so there's the 2 pieces that really help me identify this as sphingomyelin. The 5th family is glycolipid. These are pretty straightforward to identify if you feel comfortable with your sugar understanding, because what you'll see there is there's a sugar attached to this structure. And glyco is kind of short to me for sort of meaning glucose or glycogen or something along those lines, a very sugar or carbohydrate term. And lipid. So this is kind of a sugar lipid. It's a combination of both of those. And we can see out here on the right-hand side of this structure, we can see the sugar. Notice there's the oxygen in the ring structure. Notice all these OH groups, right? Remember polyhydroxylated, right? So we see all these and notice the ring structure as well. And so I definitely see a sugar group. If you see a sugar group, that's a glycolipid. But what else is a glycolipid? It looks just like sphingomyelin. It has the sphingosine backbone, shown in blue, and notice it also has, I see the amide. So if you see an amide, again, you're either going to be talking about a glycolipid or a sphingomyelin. If you don't see an amide, it's one of the other 4. But amides are found in glycolipids and in sphingomyelin. But notice the difference here is sphingomyelin is going to have a phosphate in there somewhere. Notice I don't see a phosphate at all in here, but I do see a sugar, and sphingomyelin won't have the sugar. So that'll be the big difference there. In the general structure of this molecule, notice you have, as shown here on the right-hand side, you have a big polar group up here, and then we have two long carbon tails. One comes from the fatty acid that it was attached to form that made the amide structure, and the other one is the long carbon chain from the sphingosine backbone. So again, glycolipids found in membranes as well. Uniformly, you see them in all membranes, not specific like sphingomyelin is to nerves. These are found in about every single membrane, cell membrane. So very common structures. And the sixth and final family of lipid molecules is steroids. Steroids are very different than any of the 5 classes, so these are the easiest ones to identify. They are found embedded in cell membranes, and they're also a precursor for steroid synthesis. Things like estrogen and testosterone, cholesterol is required to make those. And so although cholesterol gets a bad rap in your diet, that a lot of people say it's bad for you, it's required, though. You need it to make things like estrogen and testosterone and some other steroids that your body makes naturally. So our problem is the reason it gets a bad rap is, well, our Western diets in the U.S. typically have more cholesterol than we need. And so that's the problem. It's the excess cholesterol that we're getting, not that we're getting cholesterol. So do you need cholesterol to live? Yes, you do. It actually is very important in our vision, things like that, but it's just the excess of it is that becomes bad for you. And it starts having the problem with excess cholesterol starts building up plaques in your arteries, which start clogging, and then you start having things like heart attacks and deep vein thromboses and things like that. So it has very strong cardiovascular issues when you have high cholesterol in your diet. But we do need cholesterol for certain things. One thing that really defines this down here in the structure shown is an example, but let me just highlight a few things. Really what defines this class, or what we really see across all steroids, is these 4 rings that are fused together. Notice how they're fused. Those rings are fused. You can see 4 of them right there. And so that kind of highlights it. These other things hanging off of the ring are changed based on which steroid you're talking about. So testosterone will have those 4 fused rings. Cholesterol will have those 4 fused rings, as what's shown here. Estrogen will have those 4 fused rings. It's just the other stuff that's hanging on them will be a little bit different. Cholesterol as well has this big nonpolar region, which I'm going to go ahead and square up here for you, or somewhat circle. That big group, that is all nonpolar. Notice it's just carbons and hydrogens, that whole thing. And then we have this little polar region down here. So steroids do have polar and nonpolar regions, but the bulk of it is nonpolar, and so it acts more like a nonpolar molecule than a polar molecule because of that difference. And that's a theme through all of these. Notice every time we looked at a family, I talked about these big old nonpolar regions and these smaller polar regions. And so lipids are not really 100% nonpolar like alkanes, alkenes, and alkynes are, but they do have a little bit of polar character to them. But if you overall try to classify them as being more nonpolar or more polar, most people would say they are more nonpolar. So go ahead and pause the video, take a moment and see if you can look at these structures and try to classify them into one of the 6 lipid families. Once you think you have it, restart the video and I'll walk through that. Okay, so this one here on the left, top left, that one you can see the 4 fused rings, 1, 2, 3, and 4. And so this one is a steroid. So again, these are typically pretty easy to identify. And so this is a steroid lipid family. The one down in the middle here, kind of at the bottom, notice I see on the left-hand side, I see what looks like about 15 carbons in the row. On the other side, I see about 30 carbons. And I see this ester group right in the middle. So it looks like it has one ester group. And so when I see one ester group and an alcohol or a long carbon chain on one side and a long carbon chain on the other, this looks just like a wax to me. When you see just that one ester group and no amide structure, no phosphate, notice there's none of that. There's no sugar on this, so this is a wax. To me, this is one of the, again, probably an easier one to identify. The one on the right is pretty difficult to identify. So if you felt like you could identify this one, I think you're doing well. And so let's take a look at that. Notice I see out here, I can see what looks like a couple of fatty acid tails. I only see 2. And so that I only see 2 and not 3 fatty acid tails starts to make me lean a little bit away from triacylglycerides, because triacylglycerides would require 3 of these, and I'd have to find 3 ester bonds as well. I see 2, and so this looks like a membrane lipid because I only see 2 fatty acid tails. The question is, which membrane lipid is it, because there are 3 to choose from. I can see here what looks like an ester bond there. I see a second ester bond, and now that I've seen 2 ester bonds and I don't see a third ester bond, this is really starting to look like a glycerophospholipid. But glycerophospholipids also have a phosphate group. So just to make sure it is, I'll see if I can find the phosphate group, and I do, and I see the phosphate group right there. And so this is a glycerophospholipid. Sphingomyelin, I would have found an amide somewhere in here, and I don't see an amide. And Glycolipid, I would have also found an amide, which again I don't see an amide, and I would not have seen the phosphate, and I would have seen a sugar. And so this is definitely not a glycolipid. So just to give you a slide where it shows all the properties of fats and oils. So again, lipids, because fats and oil both are part of the lipid group of molecules. As we've been seeing throughout, we see these are basically nonpolar hydrophobic. We see that they have no ionic charges. Notice in all those molecules you never really saw, we don't really see much positive and negative charge to them. Although a couple of polar groups might have some, but for the most part they don't. Because of all that non-polar character, they're not going to be soluble, they're not going to mix well in a polar molecule with a polar molecule like water. So because these are non-polar, they don't mix well with water. And they typically are stored in adipocytes. Adipocytes is another word for our fat cells. So here shown on the right-hand side is a picture of a fat cell, a single one. And what I'm going to show you here is this is the outer edge of the fat cell. I'm going to try to get it marked up. I'm pretty close there where it is. There's a couple other cells infringing in on our picture, like up here and down here and over here. Those are just other cells that are in here, but we can almost bulkily see one cell there. And this cell has a nucleus, it has mitochondria just like all other cells do. But notice what this one has is very, very different from what you probably learned when you saw cells in some biology class or somewhere else along in your academic career or from searching on the internet. It has this big region right here in the middle that's light gray. And in an adipocyte or in a fat cell, that is triacylglycerides, or triacylglycerols. So that's where the triacylglycerols are stored for energy, that when you need energy and you don't have any other source of energy, it can use that as sort of your backup source for energy. And so notice that with this cell, how much of the cell's area or mass is really just triacylglycerides. So it's an enormous storage facility for in a cell. It's amazing how much of this space within this cell is dedicated to storing the fat, and that's stored as triacylglycerides. So it's not stored as a wax, it's not stored as steroids, it's not stored as glycerophospholipids. We store fats as triacylglycerides in these adipocytes. So this chart here you don't have to memorize, but I want you to take a couple things from it. So let's take a look at this chart. First of all, it's broken down into 2 categories, animal fat and vegetable oils. And so they show 4 different animal fats and 4 different vegetable oils. Now we all know at room temperature, animal fat these are all solids. Lard, butter. You put them at room temperature, they're not going to melt. They're going to be a solid at room temperature. We're looking at vegetable oils. olive oil, peanut oil, vegetable corn oil, those will all be liquids at room temperature. So just the state of the matter is a little bit different between them. Up here across the top, we see that these guys, try to box them, these guys are all saturated fatty acids. What this tells you, what this is showing you, is that when you talk about butter or you talk about olive oil, it's not talking about it's not made up of one specific fatty acid, but yet a combination of fatty acids. And so when we look here, notice that we have saturated fatty acids, and it shows you what percentage of each of these different sources of fatty acids, what they contain. And so across the top here you see a 12 carbon fatty acid, a 14 carbon, a 16 carbon, and an 18 carbon. And then out here to the far right shows unsaturated fatty acids. So these are going to be cis fatty acids because they're natural. And notice they have carbon lengths of, I think they both are 18. I can't read it very well on my slide. Yeah, they're both 18 carbons in length. And what we'll notice here is something, it's not so much, a couple things to note. One is, notice that they're not all made of the same combination of fatty acids. And two is there's sort of this difference between how much saturated fatty acids are in each and how much unsaturated. So if you look across the animal fats, notice, let's just take lard as an example, the first one. We can see that 41%, if we take 25, 15, and 1 from the unsaturated box, that's 41% is in the form of these three saturated fatty acids, and 56% is in the unsaturated form. So it's almost a 50-50 mix, not quite, a little bit heavier in the unsaturated, but it has a lot of saturated. If we keep going down in butter, it has 47% unsaturated. Human fat has, looks like 37% and so forth. If we go down to the oils, notice how much lower the amount of saturated fatty acids. And remember, these are probably less healthy. So these are the, I'll just term bad, but only in excess are they bad. You can still have them, but if you have heart disease, if your family has heart disease, this would be something you want to watch out for, saturated fatty acids. So the better lipids to be eating in your diet if you have concern for some of those diseases is vegetable oils because they have less of the bad fatty acids. And then these would be considered more the good stuff. So notice when you have more unsaturated fatty acids, as we do down here for vegetable oil, more unsaturated means more liquid. It's more liquid-like at room temperature. If you have more of the bad or saturated fatty acids, it tends to be more solid at room temperature. So as I talked about previously, the reason is because these so why is that? Why are some of these lipids liquid at room temperature and some solid at room temperature? And the reason is because of their structure. So if you look at a fatty acid that's saturated, and we talked about this previously, notice how, and that's the animal fats, so animal fats tend to be more saturated, and notice how that molecule is very, very linear. It's pretty straight. Where a cis unsaturated fatty acid, when you start putting double bonds into these molecules start to bend. Well, that's all fine, but still it doesn't answer the question. So let's take a look a little bit further. Now, when you start to put a couple of these together, like you'll find in the body, right? In the body, these things start to assemble together a little bit to form membranes, as an example. So think about a membrane where you have a couple of these fatty acid tails, and then it's next to another molecule with a couple fatty acid tails, and they're all sitting next to each other. When you have saturated fatty acids and the molecules are very linear, look how well they stack and touch each other. Remember van der Waals forces? Look at all that touching. A lot harder to pull apart those fatty acid tails. Think about them sticking together versus these fatty acid tails. Notice they're only sticking together in some short regions in here. The rest of them aren't sticking together. So much easier to pull apart those three on the right than the ones on the left. So it takes more energy to pull these apart, more heat to pull them apart. So at room temperature, given that amount of heat, these are less, the ones on the right, over here where we are looking at the ones with cis bends in them, those are already partially pulled apart, so therefore at room temperature that's enough heat to have moved it from the solid phase into the liquid phase. However, at room temperature, these animal fats, where they're all saturated, tend not to have been, it's still not enough energy at room temperature to pull those apart and move the molecules from the solid phase into the liquid phase. So the melting points, the MPs, the melting points of these saturated fatty acids are higher than those of the unsaturated, cis-unsaturated fatty acids. Now, trans fatty acids, trans-unsaturated, are a lot like the saturated ones because remember the trans ones are also very linear. And so trans-unsaturated and the saturated fatty acids are very similar in their melting points compared to the cis-unsaturated, and that's because of their structure. Cis-unsaturated put big bends in the chains where trans do not.